it's my privilege to now introduce Homaira Hamidzada, who is a PhD student in the Department of Immunology. She works with SLAVA and all things macrophages. Today she'll be talking to you a little bit about viral myocarditis, but I just wanted to say that I think you're just about done now, right? You're getting close to wrapping it up?
I hope so.
So Homaira, I think, has all the makings of a great scientist. She's very hardworking, incredibly bright, and when she's had enough of you, she'll tell you exactly where to go. And it doesn't matter who you are. She just walks through the lab all day long doing this. So anyway, I think that this is, I'm very, very excited actually to be able to present Homaira.
Okay, thank you, Clint, for a really great introduction and very true, and also reduced my nervousness a bit. And so I am excited today to talk to you all about macrophages in viral myocarditis. And I'd like to start with the story of a young patient whose heart suddenly fails. So they come in with chest pain, shortness of breath, arrhythmias, and the tests don't add up because these are all very general symptoms that can point to a variety of different diseases. So the cause remains a mystery. But what's interesting is that sometimes we see that patients who go on to develop dilated cardiomyopathy, we can actually detect viral genomes within the myocardium. But this is only ever seen when a biopsy is done. And this is what we call viral myocarditis, which is inflammation of the heart due to a viral infection. So now let's zoom into the heart to see how this pathogenesis unfolds. So when a virus like Coxsackie virus B infects the heart, it triggers acute myocarditis, which causes cardiomyocyte injury, immune cell infiltration, and this can either lead to reduced or preserved cardiac function. Now, in most patients, this is a self-limited process and the heart recovers, and sometimes it could or could not lead to focal fibrotic scarring. But in some patients, the inflammation persists and the heart remodels, and these patients then progress to dilated cardiomyopathy. But what's intriguing is that the reduced cardiac function in these patients is strongly correlated with the levels of viral genomes that are detected in the heart. And so this really raises the question of why is it that some patients can clear the virus and recover while others spiral into this chronic disease? It's almost like there's some hidden defense mechanisms that can protect some, but it leaves others vulnerable. And so understanding these underlying cardioprotective mechanisms is something that I've taken interest in during my PhD. And as an immunologist, I want to talk about macrophages. And in general, macrophages are involved in all stages of viral infections. But what I'd like to point out, and we've heard a little bit about this in the prior talk, is that different lineages of macrophages can possess different roles in cardiovascular diseases. So we've seen this in the context of myocardial infarctions and in hypertension. But the gap is that while macrophages are the central regulators of inflammation and repair, their functional role during viral myocarditis is unclear. And in the lab, we model viral myocarditis using the cardiotropic virus EMCV, which gives us a system to then study the functional role of these macrophages. But before getting into my overarching hypothesis, I want to give you a bit of a macrophage 101. And again, we've heard about these subsets, but I have a bit of a different nomenclature that I want to introduce to you. So in the heart, we've defined three macrophage subsets by the expression of TIMD4 and CCR2. So TIMD4 positive macrophages are the most dominant subset at steady state. They're embryonically derived and possess reparative functions. CCR2 macrophages, they're relatively minor at steady state. They're monocyte-derived and they possess inflammatory functions, while the TIMD4 CCR2 negative are more general and enriched in antigen presentation. So what I've seen is that in the context of EMCV infection, these embryonically derived TIMD4 macrophages are depleted, and instead we have this massive wave of CCR2 macrophages that flood into the heart. And so I want to focus on this elephant in the room, which are these CCR2 macrophages, and I want to point out that such a massive influx of recruited monocyte-derived macrophages in both cardiovascular diseases and other diseases in other tissues is often seen as a bad thing or something that can promote tissue injury. But I'd like to point out that monocytes are a lot smarter than we think. and that monocyte fate is very plastic. And this is because the tissue microenvironmental signals can dictate their fate. And indeed, we've sometimes seen that monocytes can even adopt fates that look like tissue-resident macrophages. And so my goal for this project was, it seems simple, but it could be a lot more complex. And so the question that I've asked is, what role or what side do these monocyte-derived macrophages take in viral myocarditis? Are they pathological, or can they actually possess cardioprotective functions? And so the model that we use to study monocyte-derived macrophages is based on CCR2, which is the chemokine that regulates egress of monocytes from the bone marrow into the bloodstream. And so when we have a CCR2 knockout mice, that prevents egress of monocytes from the bone marrow, and so it prevents the recruitment of these monocyte-derived macrophages to sites of inflammation. So in EMCV infection, I see that in the heart, during peak inflammation, we see this massive influx of the CCR2 macrophages in wild-type mice, but we have blunted influx in the CCR2 knockouts. And this correlated with increased levels of cardiac troponin, cardiac fibrosis, and reduced cardiac function, which indicated to us, this really exciting paradoxical role where recruited monocyte-derived macrophages are cardioprotective during viral myocarditis. And so the next thing that I asked is whether these macrophages can modulate the immune response during myocarditis. And what we see is that EMCV doesn't trigger much of a neutrophil response, but in the absence of monocytes in CCR2 knockout mice, we see this aggressive infiltration of neutrophils, and we also see reduced NK cells and reduced virus-specific CD8 T cells in the blood and in the heart. And so this indicated that CCR2 macrophages are not only restraining excessive recruitment of neutrophils, but they also modulate and support antiviral cellular responses. And so at this point, I wanted to gain more mechanistic insight. And so I asked the question of how the absence of recruited macrophages can reshape the cardiac transcriptome during viral myocarditis using bulk RNA-seq of total cardiac tissue. And I'd like to focus on just comparing the wild type versus the knockouts. And here I saw that the major pathways that were reduced in the CCR2 knockout mice were mitochondrial metabolism and angiogenesis, whereas I see increased in neutrophil and interferon-driven inflammatory responses. And on one hand, it was exciting to see that CCR2 macrophages can modulate such a broad range of critical pathways, but as we all know, the bulk-seq is giving us an overall global picture. And I wanted to probe cell type specific responses. So at this point, I turned to single nucleus RNA-seq, and I assessed whether we do have cell type specific responses. And what I do see is that I was able to localize to the cardiomyocyte specifically that they upregulate type 1 interferon responses and reduce mitochondrial metabolism. But I think, to me, the most interesting part of this data set personally were the fibroblasts, where I saw increased pathways related to neutrophil migration. And I'll get back to this point in just a second. I just want to recap what I've shared with you all today, which is that in viral myocarditis, we have a huge wave of monocyte-derived macrophages that enter the heart. And we can inhibit this infiltration in CCR2 knockout mice, and then we see increased cardiac injury and reduced cardiac function. We see altered antiviral cellular responses and increased neutrophils. Cardiomyocytes transition to a stress state with amplified interferon responses and reduced mitochondrial metabolism, whereas fibroblasts transition to a neutrophil attracting state. And so at this point, my working hypothesis was that the loss of monocyte-derived macrophages in CCR2 knockout mice results in an unchecked neutrophil signaling in fibroblasts promoting pathogenic accumulation of neutrophils in the heart. And so to test this hypothesis, I depleted neutrophils in CCR2 knockout mice using anti-Ly6G antibodies, and I actually saw no difference in cardiac injury, fibrosis, or function. And I'll admit, this was disappointing at first and probably not what you all thought I was leading you to, but my interpretation is that neutrophils may just be a consequence of injury, rather than the cause itself. And so looking ahead, this is a work in progress. I'm hoping to probe more deeply into the reduced mitochondrial metabolism that I've seen within the cardiomyocytes, and I'd like to see if we can rescue this cardiomyocyte metabolism using drugs that can increase or boost metabolic function. And I believe this could be clinically relevant to patients with viral cardiomyopathy. But to me, I think the most exciting next step for me is taking a spatial lens, for cardiovascular disease. Because the fact is that monocyte recruitment doesn't happen randomly. It happens within specific focal lesions in the myocardium that we miss in both bulk and single cell approaches. And so location matters, and I'd like to dive a bit deeper into spatially regulated macrophage responses in viral myocarditis. And I'll end by thanking our lab and our collaborators, and I'm happy to take questions and also any feedback or ideas. Thank you.